Will do. Some familiar faces in the audience. Thanks for the kind introduction, and thanks to DLD for the invite here. I'm actually born in Munich, so this is an opportunity to come back to my birth city after many, many decades living and working in the US, currently for SandboxAQ. And a big thank you also to the last panel. That perfectly set the stage for what I'm going to talk about, because the type of AI which was at the center of their discussion was large language models, ChatGPT, Claude, Grok. I hope to show you that there's something beyond this. LLMs are basically pattern recognition and extraction platforms, which work well if data for the question at hand is available, if they've seen things before. I would like to make the case that for drug discovery and other scientific question, material sciences, for example, battery materials, but we focus on healthcare, so no worries here. Grounding your model in physics, first principle physics, solves a couple of problems. And the fundamental limit of LLMs, as we, and I'm going to talk about who we are, but that's not the important part. The fundamental limit we see in LLMs is that they work not very well on out-of-distribution tasks. So they don't work very well on tasks where the data isn't present in the data set. So if you have a new molecule for a disease which hasn't been solved before, it struggles with assessing that molecule, the properties of the molecule, ranges from binding affinity to ADME, tox prediction. And I think the key challenge we try to solve in healthcare and in drug discovery and bringing new drugs to patients is the high failure rate in clinical phase 2. This has two major reasons. One is unexpected safety issues. The other one is poorly understood biology. And that brings us back to what do we have to do in the preclinic to actually deliver better candidates into the clinic, and how can we get there? And so, as I said, LLMs are pattern extraction machines, but molecules rely on physical interactions. So if you have a drug target, small molecule drug target, antibody interaction, it's a physical interaction which you can measure. And so the missing layer from our perspective, and that's what we have been working on over the past 4 years, are large quantitative models. Models which reason from physics and not from past patterns. So that allows us to use things we know which work, mathematical equations, quantum physical principles, and apply them to drug discovery. So we build models which are trained on fundamental laws of physics and not on past patterns. So we don't extract from the past. We do ab initio simulation and prediction of data sets based on first principle physics, and then use these data sets to train AI models. So that solves another problem. Creating data, quantitative data to train LQMs to create this in the lab, it becomes prohibitively expensive. If you try to extract quantitative data from the literature, you face a challenge that 80% of all studies in the literature are not reproducible, which also impacts the quality of LLMs, by the way. So the core principle we're following that AI, which means LLMs, extract patterns, and we use physics to constrain reality, because what we're actually trying to do in drug discovery, we try to cure real patients. And the reality around us is based on physics. So the stage I'm on is solid because of the type of interaction of electrons and orbitals, which is a quantum physical phenomenon. So the territory of LQMs ranges from protein folding, drug target binding, mRNA design, antibody optimization. And through my talk, I'm going to go from the general to the specific. But I'm not saying that LQMs are better than LLMs. The solution is actually the combination of the two. And I put the two most prominent models out there with Claude and ChatGPT. So LLMs do a fantastic job of helping us humans interact with very complex computational systems. And so that's where we see the value. LQM does very well in solving complex quantitative tasks. And that could be an affinity prediction, a potency prediction, a molecule property prediction of any kind. Combining these two also solves a big frustration we had. We built all these fantastic large quantitative models, but you needed a PhD to actually operate them because they were highly specialized. So as much as they were useful, the distribution was limited. By combining them with LLMs, and we just announced a partnership with Anthropic 3 weeks ago, we actually democratize access to the model. So now the user can call for an LQM task in Claude. Claude is intelligent enough to understand that this needs a SandboxAQ LQM model, reaches out to an MCP server, runs a calculation, and then brings the response back into the Claude environment. So the user never has to leave their familiar environment. So what's behind this? What's SandboxAQ? We spun out of Google about 4 years ago, but we're an independent company, so we're not an alphabet company. And speaking to the capital question, which was raised in the first talk, we were lucky enough to raise close to $1 billion, which gives us enough runway and allows us to tackle the hard problems. Problems where you might actually fail the first or second time and don't get it quite right. More importantly, it's a team of people. So we are a team of software engineers, mathematicians, physicists, but also all kinds of life science disciplines, computational biologists, computational chemists, medicinal chemists. And this team has learned over the past 4 years to be multilingual, to talk across the borders of disciplines, to actually build something where the engineering bias doesn't dominate the solution. So we keep our eyes on the problem and don't try to tailor the problem to what the engineers can build. And that led us to be integrated in about 20 programs across 3 modalities and indications, mainly focused on oncology, neuroscience, and autoimmune. So what differentiates us is really the focus on first principle physics, but also the proven execution across software engineering, but also science first, and operations. And our funding gives us the ability to build up very flexible collaboration models with leading academic institutions, with large pharma, with biotechs out there, even with countries to stand up their AI-driven drug discovery infrastructure. So how real is this? So let me get to real results from this approach. I'm going to pick 3 because these 3 highlight 3 different aspects of how we apply these models. The first one is Sanofi. The challenge here was Sanofi needed a mode of action biomarker which helped them to differentiate 2 different modes of action, one on the on-target and one on the off-target, which is actually part of their regulatory strategy, because if you can demonstrate that, you become first line of treatment. So there's a huge economic value attached to it. And that required building up a knowledge graph and then combining this with large quantitative models for assessing molecular interaction to deliver out of an initial list of about 9,000 candidates, 14 for which actually now patient tissue is procured, assays being developed, and the biomarkers being validated in the test to see which one of those are going to go into inclusion, exclusion criteria, and response prediction in the clinical trials. The second one is slightly unusual because it's not a therapeutic development, but it ties very well into the diagnostic representation we had in the panel before. It's a collaboration with the top diagnostic company, and I hope we can put the name to it based on the good results in the next quarter. And the challenge was they were looking for a tetravalent antibody, an antibody which binds to 4 different antigens. That's in diagnostic development a very common task because it's a cost of goods question. You want to manufacture 1 antibody for your kit and not 4. And their internal approach only gave them binders to 2 of the antigens. So we developed a tailored protein language model, which is an LLM, but then used quantitative approaches, binding predictions between the antigen and the antibody candidates to tailor the list down to about 100 clones, starting with about 20 million designs initially. And of those 90 to 100 clones, in a single shot, so in a one-shot approach, we found 3 who actually outperformed the current best binders. They bound to all 4 antigens and also at the right kinetics, which in diagnostics is crucial. You want things to go on and ideally never come off again. So last one I'm and the team are especially proud on, it's our collaboration with UCSF, with Stan Prusner's Institute on neurodegeneration, specifically Parkinson's and other neurodegenerative diseases. Stan Prusner won the Nobel Prize for finding that prions, protein aggregates, drive Parkinson's disease. It's one of the drivers, not the only one. The challenge with prions is that those are not distinct proteins. So if you think you can use AlphaFold to run a virtual screen to find binders to prions to prevent progression of Parkinson's or formation of prions, it's not going to work. We tried. They tried. What we had to do is actually recognize that prions, where proteins stack on top of each other, with a slight twist from a mathematical perspective, it's a self-replication challenge. So the solution was actually built by our mathematics group and not by our protein biochemistry group. And I think that very well reflects the diversity of disciplines you have to have in your team to tackle those hard challenges. And what we now have is called PrionDoc, which allows us to run virtual screens on prions, on vast chemical libraries to find new binders, which bind to pockets. It's going to be a long sentence. Bind to pockets, which only form when these prions form at the interfaces of it. So this is not only an example of accelerating the possible, this is making something possible which is almost impossible to execute in the lab. If you try to build assays on prions from primary patient material in the lab, these assays are very finicky, there's a high variability in there, and they're very low throughput. So UCSF was able to screen about a 2,000 compound library in a year. We can run, if you want a trillion, we run about an 8 million compound library in a week, but we can do the same for a trillion compounds. I don't think that's necessary from a chemical space perspective, but again, this is an example where we not only accelerate things, but where we open up a therapeutic target space, which is almost impossible to interrogate efficiently in a wet lab approach. So what's our vision to drive this forward when physics meets AI at scale? So what we've seen and what we've shown in our partnerships is that we are able to design more precisely binding drugs using physical approaches than, let's say, with co-folding approaches alone, with an AlphaFold approach. And we can do this before they're even ever synthesized. So that brings down the cost, accelerates the timeline, but more importantly, gives us hopefully better drug candidates, because with this approach, we can very efficiently also look for what is the binding on the on target. That's what everybody does. What's the binding on the off target? And if you combine this with our knowledge graph approach, we can also look at other synergistic pathways we also need to hit, because our bodies or a tumor are very good at, even if we hit our primary target, they find a way around it, which is a big cause for lack of efficacy in clinical studies. The second point, rare diseases, goes back to my initial point. What do you do if you do not have data or your amount of data is very limited? And that applies especially to rare diseases. The amount of data is limited. The variability within the data is high. So it's the perfect application for LQMs, where you take an ab initio approach and don't look at pre-existing data, but simulate the relevant data from scratch. Precision medicine, not going to go into detail here. It's the same kind of logical approach. But I think one of the most important aspects is really the combination of LLMs and LQMs to democratize access to these highly sophisticated large quantitative models to a much broader user base. So to take this limitation away, that you need a fairly sophisticated educational academic education level to actually operate them. So what I hope I can leave you with is that the question is not if AI will transform medicine. It is already doing it, and it will continue doing it, but the question is, are we using the right kind of AI to the fullest extent to solve some of the hardest problems? Furthermore, that large quantitative models are the missing layer between the LLMs, where we can naturally interact with and communicate with, and some of the quantitative questions which leads us to the precision drug discovery needs. Then finally, that we at Sandbox build this infrastructure. We have the team, we have the funding, we have the vision and the energy to go after the really hard problems. So if you are looking for partners in this space, we are happy to discuss. So please reach out to me. Thank you for your attention.